Assalamu alaikum students. Hope so you all will be fine. In this video, I am going to discuss about Chylomicron metabolism. We will cover this topic under the heading of need of chylomicron, function, structure, metabolism and disorder. So start our lecture. Why there is a need of chylomicron? Students, as we know that lipids are insoluble in water. So, for their storage, transport, and utilization, they form complex with protein to overcome this problem. So that's why there is a need of synthesis of chylomicron. Let's see. Triacylglycerol, cholesterol ester, phospholipid, cholesterol, and protein. These lipids they form complex by attaching with themselves with a protein and they form lipoprotein. Students, triacylglycerol and cholesterol ester, these are non-polar lipid. Phospholipid and cholesterol, these are amphipathic lipid, mean hydrophobic and as well as hydrophilic. So why there is a need of chylomicron? Because lipids are insoluble in water. So for their storage, transport and utilization, they form complex with the protein. So that's why. Now function. Chylomicron is responsible for transport of exogenous triacylglycerol from gastrointestinal tract to peripheral tissues. This is the function of chylomicron. Now coming to the structure of chylomicron. Structure of chylomicron. This consists of two layers. Here is a phospholipid, only one layer of phospholipid. Phospholipid. There is also cholesterol. It is a cholesterol. There is also peripheral protein. It is a peripheral protein. This is a integral protein. There is a triacylglycerol cholesterol ester. So student, this is the structure of chylomicron It consists of two layers. At the outer layer, there is only phospholipid, only one layer, cholesterol, and peripheral protein, integral protein, triglycerol, and cholesterol ester. Now coming to the metabolism of chylomicron. Metabolism, there are some steps you must understand to understand the chylomicron metabolism. So must remember these four steps of chylomicron metabolism. First one is nascent. Sorry. First one is nascent chylomicron formation. Second one is maturation. Third one is chylomicron remnant formation, and fourth one is uptake. Students, these are the four steps in the chylomicron metabolism: nascent chylomicron formation, maturation, chylomicron remnant formation, and uptake. Now coming to these steps. Students, the synthesis of chylomicron takes place in intestine. This is intestine. Basically, how the synthesis of chylomicron takes place. There is a synthesis of Apo CIII protein in rough endoplasmic reticulum. There is a synthesis of lipoprotein in smooth endoplasmic reticulum. And this Apo CIII, it gets incorporated in lipoprotein. In smooth endoplasmic reticulum, there is also synthesis of triacylglycerol. In Golgi bodies, further groups, carbohydrate groups, they get attached to this complex, and this complex gets incorporated in chylomicron. Through reverse pinocytosis, they are transported into lymphatics. With the help of thoracic duct, they are transported into the blood. Okay, students, I am repeating again. In rough endoplasmic reticulum, there is synthesis of Apo CIII protein. In smooth endoplasmic reticulum, there is synthesis of lipoprotein and triacylglycerol. Okay, and Apo CIII, it gets incorporated in lipoprotein. In Golgi bodies, further the carbohydrate group gets attached, and this complex gets incorporated in form of chylomicron, and through reverse pinocytosis they are transported into lymphatics, and through thoracic duct they are transported into the blood. Here is a nascent chylomicron formation. With the help of lymph, they are transported into the blood. Nascent chylomicron formation. Triacylglycerol, cholesterol ester, there is Apo CIII and apoprotein. This is the nascent chylomicron. In next step, Apo C2 and Apo E, they gets incorporated in nascent chylomicron. Apo C2 and ApoE, they are obtained from HDL. Apo C2 and ApoE from HDL gets incorporated in nascent chylomicron and it leads to the maturation of maturation of chylomicron. Apo B48, Apo A, Apo E, Apo C2. This is the mature chylomicron. In next step, students, lipoprotein lipase enzyme which is present on capillary wall. It gets attached to the endothelium surface by heparin sulfate. This lipoprotein lipase acts upon triacylglycerol and responsible for the hydrolysis of triacylglycerol in peripheral tissues. Lipoprotein lipase. Lipoprotein lipase enzyme which is present on capillary wall which is fixed to the surface of endothelium by heparin sulfate. This lipoprotein lipase enzyme it is responsible for the hydrolysis of triacylglycerol and fatty acid they gets transported into the peripheral tissue while there is a glycerol. Lipoprotein lipase enzyme it is activated by Apo C2 protein and it is inhibited by Apo A2 and Apo C3 protein. Students, I will repeat it again. This lipoprotein lipase enzyme which is present on the capillary wall which is fixed to the surface of endothelium by heparin sulfate. This acts upon triacylglycerol molecule and converts it into glycerol and fatty acid and fat is transported into peripheral tissues. This lipoprotein lipase enzyme activated by ApoC2 protein and inhibited by ApoA2 and ApoC3 protein. This lipoprotein lipase enzyme, here is a role of hormone insulin. Insulin, it is responsible for the increase in synthesis of this lipoprotein lipase enzyme and responsible for the translocation of lipoprotein lipase to the surface of endothelium. Now 70 to 90%, 70 to 80% triacylglycerol it gets hydrolyzed by lipoprotein lipase enzyme and it leads to the reduction in the size of mature chylomicron. Triacylglycerol, cholesterol ester, ApoB48, here is a apoprotein and it loses Apo C2 and Apo it loses Apo C2 and Apo A protein. Sorry, here is the Apo A, Apo E. Okay, students, it loses Apo C2 protein and Apo A protein, and here is the formation of chylomicron remnant. Three steps are completed. Okay, students, nascent chylomicron formation, mature chylomicron, and here is the chylomicron remnant formation. Four steps are completed. This is the first step, second step, this is the third step. In next step, this chylomicron remnant is uptake by liver. Okay students, and it is done by receptor. There are two types of receptor that are present on liver for the uptake of chylomicron remnant. LDL receptor and LDL receptor mediated protein. Hepatic lipase enzyme, it is a very important enzyme which is present on the surface of liver. It is responsible for the hydrolysis of triglycerol and it is responsible for the uptake of this chylomicron remnant. Okay students, this in this way this fourth step is completed. So students, this is the all about chylomicron metabolism. Four steps are there: nascent chylomicron formation, mature chylomicron formation, third one is chylomicron remnant formation, and fourth one is uptake by liver. This is all about the chylomicron metabolism. Now coming to the very important disorder, its name is type 1 hyperlipoproteinemia. Type 1 hyperlipoproteinemia. It is due to the deficiency of lipoprotein lipase enzyme. It causes the increasing level of triglycerol in the blood. This is the disorder type 1 hyperlipoproteinemia. Okay, students, this was all about the chylomicron metabolism. Keep watching and thank you.